Yes, I feel a bit of a fraud after John's talk because it was some great science there. And this is my slide. So don't expect it to change because it won't. You're going to have to look at me, I'm afraid. So I just wanted to sort of take on a little bit of a journey to just explain what we've been doing at Discuva and how we've been changing hearts and minds, I guess, as we've been developing our business. So the whole story started in 2009. I was at a social function and I met this guy called John Wayne, who was running one of the major reference laboratories down at Colindale, looking at microbial infections. And he said to me, David, you're from the pharma industry, we need some more antibiotics. I mean, that threw me because I wanted a burger and he was cooking on the barbecue. So it was, but I let that go and we kept on talking. And the interesting thing was that we both had something to contribute. So he was working on some pretty neat technology, which was good, but it wasn't what we could use for drug discovery. And I was working on some antibiotics in a company I was at. So it was, there was something there. There was a seed of something there. So we decided to sort of pool our resources and try and make a difference to antibiotics and developing new ones. So there were two things we needed to do here at the beginning of the company. One was the market research. We had to find out whether anyone needed any new antibiotics and what were the problems in the industry, and also how do you sell into that. The other thing we needed was a really good team. I mean, that was absolutely crucial. So taking the market research, what we did was we essentially went to everyone in the world that was developing or discovering new antibiotics and said, well, what's the problem? What's the problem with in your company today? What do you need to solve? And the big issue was getting chemicals into bacteria, especially the drug-resistant bacteria that we have in our hospitals at the moment. The gram-negative bacteria are just almost untreatable now in many cases. So there were sort of two ways you could sort of get molecules in. You could change the chemistry, or you could look at things that could actually stop bacteria growing and then sort of find out how those things worked, those molecules worked. But the technology wasn't there to do that. So this is phenotypic screening. This is being able to screen against huge collections of compounds, and once you get something that either slows down the growth of a bug or kills it, actually find out what that is, what that's doing, what the target is. So we thought, oh, hang on, that's good. There's a real need there. We'll work on that. And John's group had the seeds of a technology that could be used to this, but they weren't good enough. So what we had to do was get a team to work on that to make it better. So Cambridge is brilliant, because you can just look out the window and find someone who's an expert in something, and you probably know them. And that's exactly what we did. So we went to our network, and we got a really good geneticist, we got a really good screening person, we got a really good chemist into the team, plus we got an extremely good SAB together. We got the thought leaders in the area together, and all working for nothing, which is great. And we started working on it, and we started working on the problem. And it took us about a year and a half to develop the technology in thought, but we filed the IP and decided to go on the road and gave up jobs and stuff and started treading the pavements. So we managed to raise some money, and at the end of 2011, December, we got some money in the bank, and we went up to one of the science parks and did the quickest lease ever. 2.5 weeks, we were in the labs just before Christmas. So over Christmas, we bought the equipment, moved in on the 3rd of January, started working. Validated the technology in 5 months, completely from scratch. So the technology, right, so what is it? Well, what it does is we make these enormous libraries of bacteria. So if it's a drug-resistant bacteria, let's just take E. coli, that's one, we work on 5 or 6. Make an enormous library of different mutants. And the mutants have special functions, so we can turn on genes, we can turn off genes, we can do all sorts of neat things to genes. But the density of these mutations is so great. So every single member of this library has a mutation in a different place. And the different place varies by as little as 4 base pairs. So you literally cover the genome in this library with all these different mutations. And you get these bacteria, and you throw them in with a compound that you want to know what's happening with. And you've got the guys at the front, the mutants are going, you've got the mutants that come out. What we do is we sequence everything. So we sequence the beginning, sequence the end, and compare the results. And from that information, you find out everything you want to know about the bacteria. You find out all it needs to grow, so all the essential genes in that bacteria. You find out the target of your compound. You find out any gene that's going to result in resistance through the clinic with your compound, potentially. So that's the technology. We've validated it totally. In growing the company, we had to hire other people. So we had to bring in bioinformatics. We had to bring in next-generation sequencing expertise. It took just a couple of weeks in each case to hire these people through our networks and through the amazing amount of expertise you have in Cambridge. So the technology now is working. We're screening molecules. So we've managed to acquire, through various means, huge libraries of compounds that we've selected for various physicochemical characteristics. So within the company, we've built high-throughput screening, genetics, bioinformatics, and all the library building capabilities, all in-house, and next-generation sequencing. So we start screening, and currently up to date, we have about 3,000 different new antibiotic scaffolds that we've identified, all new, which work in gram-negative bacteria. We've raised funding to pursue 12 of these through Hit to Lead to proof of concept. So we've done an A-series round altogether with both our investors and also the TSB, who've been very helpful. So these are good. We've taken these through to some chemistry, and we've now got them down to potencies, which is drug-like. We've got a bit of work to do. We've got to tinker with some in vivo properties of these molecules. So we have 2 series down at that level, and we have another 10 series that are currently going into chemistry or in chemistry at the moment. So that's in a year and a half. So that's with the right team in place. But what we've had to do as well is we've had to change perceptions, because I don't know if anyone's from the anti-infective community here, but it was a little bit underinvested. A lot of pharma got out in the 90s thinking that antibiotics were no longer needed. And so there was a lot of dogma in the area. And a lot of people thinking, well, OK, we just need a few more quite broad-spectrum antibiotics. And we believe very strongly that that's not the way forward, because if you develop a broad-spectrum antibiotic, it's likely to lead very quickly to resistance. So what we were proposing was more selective therapies. Therapies that are disease-specific. So just treat the disease and the bacteria that cause the disease. And doing that, you can discover new targets, because these are targets that are specific for certain bacteria or certain diseases. So resistance potentially goes away, brings new chemotypes. But also it's a sort of, there was a perception that you needed a drug that could hit multiple targets inside a bacteria as well, which is absolute insanity when you're trying to discover drugs and optimize for a particular target. But that attitude was out there. So what it said was, well, OK, if you want to hit multiple targets in a bacteria, why not just try 2 drugs, 3 drugs, same as what we do in HIV therapy, for example, where resistance goes away with combination therapy. So these were the concepts that we had to sort of seed out there in the industry. And so we started doing that as we were raising money, actually. So we did that in a number of ways, talking to key opinion leaders, but also we organised a conference. And we got together clinicians, regulators, and people from NICE, biotech, pharma, got them in a room in Cambridge and got them talking. So started to seed all these sort of ideas. And it's quite interesting, actually, because when we started out on this journey, the perception was we need broad-spectrum antibiotics and we have to save them until we need them. So commercially, it wasn't viable to anyone to develop new antibiotics. What we wanted to do was say, look, there's new models out there that make this a viable proposition in terms of developing new antibiotics. So they were the seeds. And at the G8 conference in June, this is why they're changing the world things there, the overriding conclusion at the summit in London was that what we needed was new chemical entities that were more selective against specific bacteria. So I think we're sort of winning. I don't know if it's us that did that, but certainly the world's changing, and we're in the right place at the right time, I think. And that's my talk. I don't know how long that was. One plug. One plug. So the second slide. We've got another one bug, one drug in September. So if you want to come, just look at the details there. Thank you.
Hi, Miles Carroll from Water Health Protection Agency, now Public Health England.
Yep.
I don't think Big Pharma got out of antibiotic drug development because it didn't think there was a need, because it was too risky and bacteria were becoming resistant too quickly, and it wasn't financially worth their while. And so have you shown that your technology can identify targets where bacteria cannot become resistant overnight, as with our present situation?
I think so. We've got some targets that look pretty interesting. So we have got new targets. We've got new chemotypes against new targets. It's early in the platform, in the drug discovery programs, but the technology has been extremely effective at predicting resistance. So, for example, from our platform, which we call Satin, we've been able to say, look, these are going to be the resistance genes to this particular chemotype. And then we've been able to take that chemotype and actually just generate resistance by incubating it at higher and higher concentrations. And we've literally shown that those are the genes that do come up as resistance genes. So it works very well. It works very quick, 3 days, the whole process, 3 days, very quick. So it's in the drug discovery pipeline, cycle times. So I think that gives us the confidence to know that when we select a drug, when we do our triage, which is, we get all these hits, we say, well, which ones are worth pursuing? Well, we can't get IP on that. That one's a single target. That one hits a gene at the end of a pathway. That gives us that, this only has 2 resistance genes that might be a problem. This one doesn't have any that are coming up at the moment. And that really is giving us confidence. The platform works very, very well. It's very predictive. So I think we're onto a winner. They are, yes. So,
sorry, can you just repeat the question?
Sure.
Sorry.
The mutation. Can everyone hear me? The mutations that you generate, are they reflective of what happens in vivo to bacteria? You said yes.
Yeah, they are. And I mean, the technology. So, for example, with we have overexpression transposons that we use, for example, and that'll bring up things like efflux pumps, transporters, and enzymes within the cell that actually degrade certain scaffolds. And all of those genes come up in vivo in natural situations, absolutely, yes.
Yes, please. Just a microphone coming, sorry.
Julie Pratt, University of Leicester. I just wanted to ask about your... Luke's got my question. Okay, I'll ask a second question.
Sorry.
I was in mid-flow there.
Yeah.
Okay, well, first of all, I suppose I wanted to just say that drugs that hit multiple targets are, an example of that would be penicillin. And so I think that is quite a good antibiotic, talking from the drug discovery process for antibacterial drug discovery. But the other thing I wanted to say is that, I remember the question now. These new scaffolds and things and new drugs that you said are completely new entities. Are they natural products? What's the sort of cost of goods relationship with that? I mean, are they actually going to be feasible as drugs in the future?
They absolutely are. I mean, we have a collection of something like 70,000 molecules at the moment, and that's expanding all the time. Very small proportion of that, we said, well, let's look at some natural products. Very small proportion, about, 1,000 natural pure compounds. Got loads of hits there, but we really we're really pursuing the things that have come from proprietary, that have come from sort of vendor supplies. So they're small molecules, they're very, very cheap cost of goods. The sort of number of steps of synthesis is something like between 4 and 5, 6, that sort of level in terms of, getting to your lead molecule. So they're going to be very effective to produce a very cheap price. Coming back to your first point, yeah, we've tried some of the beta-lactams through the platform, and you're right, they do hit different things. But what's very interesting, we've got an enormous amount of data that you guys that we'd love to share. And with the beta-lactams, we find that, yes, you can get them hitting multiple penicillin-binding proteins, but it's the ones that are essential for growth that are the ones you're really interested in. For example, we did an experiment recently with imipenem. There's one specific penicillin-binding protein in the bacteria we were looking at the time that's a key target in that bacteria. So I sort of agree with you, but I don't agree. I think if you're going to hit something in different pathways, which is probably the best thing to do to stop resistance developing, you need 2 molecules.
Very diplomatic answer, David. Good. There's time for one more question if there's one out there. If not, then we'll move on. Anybody else?
Can I ask one? I can shout loudly enough, I don't need the microphone.
The big issue that I've heard from pharma is not that there's no interest in developing new antibiotics, it's pricing. Because the expectation, it's very difficult to charge a premium for a new antibiotic. You're working on a premium to a generic, and that's actually not commercially very attractive. So that's what I've heard. So the issue is the pricing of the antibiotic, not the new therapy.
I mean, that certainly was the case a couple of years ago, and you will see that a lot of pharma that got out are now coming back in, and if they've not got back in, they're telling their corporates to invest in that area now. And one of the reasons for that is, well, once you get 5% resistance to something, a clinician is not going to give you a generic. So there is a need for new drugs. The other thing is the regulatory authorities are changing the way they look at new drugs now. So, for example, with antibiotics now, there's a thing called Tier C, which is something, it's a concept which has been agreed, well, which is being agreed by the EMA and the FDA. It's been supported by the insurance companies in the US, where you can go straight from phase 2 to named patient, limited number of patients, but for £30,000 per treatment. It's really looking at the whole cost economics of the problem, and if someone's in hospital and they're going to be there for a long time, it's much more cost effective to give them a treatment that gets them back home. I guess the current only real selective medicine we've got at the moment is fidaxomicin for C. diff. And that's a bit of an experiment at the moment. It's sort of leading the way. I know it's about $2,800 per treatment. And some hospitals prescribe it and some don't. And I recommend use of it. But so that's one. But the whole thing's moving. And the resistance problem is huge. I mean, you talk to any clinician anywhere in the world, and they're experimenting with current medicines. I saw a recent talk where a clinician was risking 40% chance of knocking out someone's kidney or hearing just because they were going to die otherwise. And that's what we're up against. So there is a real clinical need.
Okay, good.
Yep.
Thanks very much, David.
You're welcome.